Ordinary cell division in plants and animals, for that matter, is referred to as mitosis. And the steps of mitosis we'll talk about in detail in the next section. But before we do, we need to talk about the most important factor in mitosis, and that is making sure that the offspring cells or the resulting cells from the original cell have an exact copy of the so-called owner's manual or genome of the original cell. So that owner's manual has to be meticulously copied and distributed to each of the new cells. And so we need to look at the form of that owner's manual. And you might recall that we said in earlier parts of the class that this manual is written in the language of DNA. And DNA is further divided into what are called chromosomes. And a chromosome is just a discrete piece or one single piece of DNA. And the DNA, again, must be copied completely before a cell divides. Otherwise, you're going to have mutant cells being created. They won't have a complete copy of the owner's manual, and so they therefore likely will not be able to function as a normal cell would function. How does a plant cell accomplish this? Well, it again has its DNA or its entire owner's manual broken up into, you could think of them as books or chapters, let's say, referred to as chromosomes. When we look at a nucleus, and we've drawn them in the past, and you often see basically it's pretty nondescript when you look at it. It's just kind of grainy looking in past photos that we've got in the handouts. You really can't see anything specific inside of the nucleus. And the chromosomes are in there, but in the day-to-day operation of a cell, the information on those chromosomes is constantly being copied and read and used to make what we call transcripts of RNA, or which are expendable copies of the DNA, to protect it. Remember, we referred to this as the reference library of the owner's manual, and copies can be made and sent elsewhere in the cell, but for the most part it protects the reference copy in the nucleus. And so most of the time, if we look through a microscope at a cell, we can't see individual chromosomes. However, prior to dividing, these chromosomes have to get much more organized or much more condensed so that when the cell divides, none of them get tangled up or none of them get broken in the process. And so before a cell divides, these chromosomes, which are just pieces of DNA, start to condense. And they condense, well, we've seen in this class, I know before, and you're probably familiar also with the double helix, as they call it, of DNA. In this form, it's just all stretched out. Before it can divide, again, it has to condense it. And so these strands, instead of being stretched out, okay, they start to coil primarily, and then they'll coil a second time secondarily, and there are proteins around which these pieces of DNA or these chromosomes will coil very, very tightly, much like coiling thread on a spool. As they condense, they start to become more visible when we look at a cell, and eventually they start to look something like this. And this is what you commonly see when you see a figure or a drawing or maybe even a photograph of a chromosome. I'm going to add this little white portion here. I'll talk about the different parts here. Now, this is also in your notes. There's a figure in your notes. These arms of the chromosome are referred to as chromatids, and so one of these is a chromatid, and so is that one. And because they are connected in the center and they are part of the same chromosome, they're referred to as sister chromatids, plural. And this sister chromatid on the right-hand side is an exact copy of the sister chromatid on the left-hand side. Now, these chromatids are connected in the middle by a structure known as the centromere. And it's basically some proteins, and they kind of act like a glue, and keep the sister chromatids stuck together before it goes into full-blown cell division. So the chromatids are the strands on either side of the centromere, and the centromere are the thick centers of a chromosome. And this is the way a chromosome will appear, and you can actually see this under a light microscope during the initial phases of mitosis or cell division. In fact, let's take a look at one. This photo shows chromosomes clearly visible prior to cell division. And if we look closely, we can sort of see a X-shaped, I'm going to try and trace it here, X-shaped chromosome here. And we can see its 4 arms. And it's connected in the middle by a centromere. Again, they have to be condensed down to this form so that when they get pulled to either end of the cell, as we can see has already happened in this cell that's next to it, they don't become tangled and broken off and basically lose part of the owner's manual. And so when this cell later divides in half right in the middle, each will have in its nucleus a full copy of the owner's manual. Next, we're going to be talking about the various steps in this process, and it's referred to as the phases of mitosis.